in my undergrad, I was at Caltech and I was working in Eric Davidson's lab, who's considered in many ways the grandfather of gene regulatory networks. And we were working on gene regulatory networks in early sea urchin development to understand how these connections of genes regulate development using a model organism of the sea urchin. And I was really excited about how we can understand the architecture of the gene network to understand these aspects of development, and was really interested in how we might be able to apply this to understand human development, as well as how it goes awry in disease. And I had the opportunity to pursue that during my MD-PhD work here at Gladstone and UCSF, where I applied these concepts to develop a framework where we could use machine learning and stem cell modeling to first map the network of genes that was disrupted in disease. And then, having understood that map, understanding what's central in that network, screen for drugs that could correct the network back to normal, as opposed to just treating something that is not central but more peripheral in the network that might treat symptoms but doesn't really target the underlying disease process. And doing that, we were able to identify a candidate therapy for a disease of the heart valves that was able to correct the network back to normal, and also showed efficacy both in the stem cell models we were using as well as in vivo in the mouse model, and is now under further investigation towards clinical trial.